So I did my presentation on smell and taste, and I'm going to be skipping a few slides and summarizing a lot of the slides just to keep the time around 5 minutes. So gustation is our sense of taste, and taste receptor cells are called gustatory cells, and they are found in special sensory organs called taste buds, which are found on our tongue. Papillae are elevations of epithelial and connective tissue on the tongue, and there are 4 different types. The first type is filiform, which has no taste buds and detect texture of food. The second is fungiform, which contain a few taste buds. The third is vallate, which are the largest, and they are surrounded by deep depressions where most taste buds are found. And lastly, folate, which are not well developed on the human tongue, but they do contain a few taste buds during childhood. This is a diagram that summarizes the structure and location of papillae on the tongue. Taste buds are composed of gustatory cells enclosed by supporting cells, and the dendritic endings of gustatory cells form the microvilli, and they are the receptor portion of the cell that chemicals stimulate. Gustatory cells are replaced every 7 to 10 days by stem cells known as basal cells. As we age, beginning around age 50, this replacement slows down, leading to reduced ability to distinguish tastes. Our tongue detects 5 basic taste sensations. They are sweet, salt, sour, bitter, and umami. In order for any of these tastes to be sensed, a threshold must be reached for stimulation. The threshold required varies for each taste, and actually our taste buds are more sensitive to bitter and sour stimuli because they are indicative of something that may be toxic or poisonous. Now I'm going to talk about olfaction, which is our sense of smell, and the process functions by odorants, which are volatile molecules entering into the nasal cavity, and these molecules must be dissolved in order to be detected. There are numerous components involved in the olfaction process. These include the olfactory epithelium, which lies in the superior region of the nasal cavity, and the olfactory epithelium is composed of 3 primary cell types: olfactory receptor cells, supporting cells, and basal cells. The olfactory receptor cells are regenerated every 40 to 60 days and regenerate within the olfactory epithelium. Inside this epithelium is a connective layer called the lamina propria that secretes mucus, which allows odors to dissolve so they can ultimately be detected. As we age, the mucus production slows, and as a result, the effectiveness of our sense of smell deteriorates. The olfactory receptor cells are one of the most important components of the olfactory organ system, as they are essential for detecting odors. These cells are specialized sensory bipolar neurons that bind odor molecules. From an anatomical perspective, the region in which the odor molecules bind to the receptor cell is seen as a thin knob projection containing olfactory hairs. These hairs are cilia-like extensions and are both unmyelated and immobile. They are where the odor molecules bind to activate the receptor cell to begin the production of a nerve impulse. This nerve impulse helps our brain determine what smell is being detected. How do these receptors distinguish between different odor molecules in order to convey the right message to the brain? Well, this is done because the olfactory hairs contain different receptors for different odors. Therefore, the olfactory receptor cells, which are neurons, can transmit a different signal, i.e. a different smell, depending on what receptor is being activated. After these receptors are activated, rapid adaptation occurs, and the receptors become desensitized to the odor molecules. This is why potent smells such as rotten garbage becomes more bearable to the nose after a few minutes of being around it. Lastly, an important note about the function of the receptor cell is that in order for odor molecules to bind and activate the receptor, they must first be dissolved in the mucus. Lastly, the olfactory system has a higher recognition capability compared to our taste system. It can recognize primary odors and secondary odors. The primary odors include fishy, musky, and sweaty, whereas the secondary odors are made up of thousands of chemical stimuli. To finish off the presentation are two questions, one based on the presentation and the other is more of a fun fact. So question 1 is, what are the 4 types of papillae and where are they located on the tongue? I didn't really talk about the location, so knowing what the 4 types are is more important. And the second question is, how many different smells can a human detect? I will give you a second to think about the possible answers. So the answer to question 1 is filiform, fungiform, phthalate, and folate. And the answer to question 2 is the human nose has roughly 400 types of sense receptors that can detect at least 1 trillion different odors. The human nose can distinguish at least 1 trillion different odors, a resolution orders of magnitude beyond the previous estimation, which was 10,000 cents. So this is the end of my presentation, and thank you for listening.